Hi, I'm Tanya Siddiqi at City of Hope Medical Center in California, and I presented the primary analysis data from TRANSCEND CLL004 study for the first time at ASCO 2023, and then again at ICML in Lugano this year. This study is a phase 1/2 open-label multi-center study of lysocell CAR T cells in relapsed refractory CLL patients. And the reason why we chose to even do this study is because there are a subgroup of patients in CLL who are emerging and have a high unmet medical need. And those are patients who are progressing now on our new BTK inhibitors as well as venetoclax treatments. So you know that CLL is an incurable type of a cancer or low-grade lymphoma, and we've come a long way in the treatment landscape of this disease, and it's all sort of now non-chemo targeted therapy options. But when patients progress on these novel agents, there's not a lot we can offer them reliably. And so CAR T cells might help, provide some kind of durable remissions in this patient population. And so we enrolled a total of 137 patients. They were all leukapheresed. Bridging therapy was allowed if needed while CAR T cells were being manufactured. Our manufacturing rate was 96%. It was great. And then after restaging, everybody received their fludarabine and cyclophosphamide lymphodepletion therapy, followed a couple of days later by the lysocell CAR T cells. And we've presented on and published previously the phase 1 portion of the data. So we know that dose level 2 is the recommended phase 2 dose of 100 million cells. And so now, for the first time, we presented and published at the same time and in Lancet the data from all the patients who were treated at dose level 2, the 100 million CAR positive T cells. And to be eligible for this study, patients needed to have failed at least 2 or 3 prior lines of therapy for CLL, depending on whether or not they were high-risk CLL or not. And everybody needed to have had prior BTK inhibitor therapy to be eligible. We did have certain primary and key secondary endpoints that were tested statistically in a hierarchical manner. So starting with complete remission rate, our null hypothesis was less than or equal to 5%. Overall response rate, null hypothesis was less than or equal to 40%. And then thirdly, undetectable minimal residual disease to a level of 10 to the minus 4 in the blood. We did it by next generation sequencing, clonoSEQ assay, sort of retroactively or retrospectively. And our hypothesis, null hypothesis, was less than or equal to 5% for that. We did have a pre-specified subset of interest, which were the patients who had not just progressed on BTK inhibitor therapy, but also failed to have an adequate response to venetoclax therapy as well. And so if you look at the consort diagram, out of the 137 patients who were leukapheresed, 117 were actually treated with their lysocell CAR T cells, and about 96 were evaluable for efficacy. Out of those, 87 were on the dose level 2, which is our dose level of interest. And on the right side, we show that subset of interest, which are the double refractory, if you will, population who progressed on BTKI and failed venetoclax in the past. And that subgroup of efficacy evaluable patients in this smaller population was 49 at dose level 2, and those are the patients where the primary endpoint was analyzed initially. And so our study primary endpoint of independent review committee assessed complete remission rate was met at 18% in this smaller subset of double refractory patients of 49. It was highly statistically significant compared to our null hypothesis of less than or equal to 5%. And it is very equivalent to the rest of the population treated at this dose level in this study. Overall response rate was 43%. That unfortunately was not statistically significant, so we could not formally statistically test the hypothesis in the next category, which was UMRD, but the UMRD rates were very high at 63% in the blood and 59% in the marrow. So there was a high concordance between the two compartments. And all these results are very similar in the rest of the patient population outside of this double refractory subgroup. The breakdown of the overall responses are shown below. And I'd like to mention that the median time to first response was very quick at 1.2 months. Also, the first time point of 30-day disease assessment after CAR T cells, pretty much all the UMRDs were already there. You know, they happened very quickly. So it was rapid and deep remissions. Pretty much all the PRs and CRs were UMRD. There were also 12 out of 20 evaluable stable disease patients who were UMRD by this time. I'd like to just point out that venetoclax failure in our subset is defined as patients who had either progressed on venetoclax, they had stopped venetoclax for intolerability, or they had not had an objective response within the first 3 months of starting venetoclax. Because venetoclax works very quickly, we feel that if they don't respond within the first 3 months, they're probably going to fail it or are failing it already. So here's Kaplan-Meier curves for duration of response by best overall response. On the right side, we show the smaller double refractory patient population, or the primary efficacy analysis subset, I should say, of 49 patients. You can see that the median duration response in the CR patients in blue had not been reached at this median follow-up of 19.7 months. And the median duration response for all those patients in that group was 35.3, very equivalent to the rest of the patient population on the left. If you look at progression-free survival by best overall response and MRD, so on the right again is the primary efficacy analysis subset of 49 patients, you can see that median progression-free survival for the CR patients in blue has not been reached, and for the PR patients is 26.2 months. Pretty much all of them were UMRD. That's why it's the same for PFS for PR patients, whether or not you look at the UMRD curve or not. And then we start to see that in the stable disease patients in red and green, the UMRD patients had a little bit better progression-free survival median at 6.4 months compared to the detectable MRD patients in green. And it's fairly similar in the rest of the patient population on the left. And so we think that the single administration of liso-cel demonstrated fairly deep, rapid and durable responses in relapsed refractory CLL. You know that these patients are multiply refractory. The median prior lines of therapy was 5 in this patient population. You know, a majority of them had failed both BTKI and venetoclax. And we also wanted to point out that the primary endpoint of complete remission rate was met at 18%, not just in our subset of primary efficacy analysis patients, but also in the rest of the efficacy evaluable patient population. We did see very high UMRD rates in blood and marrow. And I didn't show you this here, but in the presentations and publications, we do show that the CAR T cells manufactured were functional. They demonstrated expansion and persistence in pretty much everybody, and expansion did track with responses. The safety profile was quite manageable. There were primarily only grade 1 and 2 CRS and neurotoxicity events. There was no grade 4 or 5 CRS event. There was 1 grade 4 neurotoxicity event, no grade 5. And for the most part, you know, the liso-cel was fairly safe and manageable in these patients. And so we feel that these results support the use of liso-cel as a potential new treatment option for relapsed refractory CLL patients, especially those who have progressed after BTK inhibitor therapy, and maybe even those who are the double refractory population after BTKI and venetoclax therapy. My hope is that we get approval for this treatment regimen by the FDA at some point soon, and that so that we can offer it to patients not after 5 prior lines of therapy like we did for this study, but maybe sooner, like really after 2 prior lines of therapy. And then in the future, I think we need to study this even in earlier lines of therapy for patients who maybe have high-risk CLL, maybe after just 1 prior line of therapy, but that's all sort of future planning and ideas that we need to vet, because certainly we do need to improve upon these results, but this is, I think, an excellent first starting point for patients who have fairly refractory CLL and not a lot of great options left to them. One-time treatment, you watch the patients closely for a month, and then I've had patients now 3 to 5 years out who haven't relapsed and are living very normal and good quality of life, where they don't even need to come see the doctor very often, which is great. So there's a lot of clinical benefit that we found in these patients. I usually like to point out that, if patients and doctors can come in to see, us at transplant and CAR T centers early, maybe after they've just progressed after the first novel agent, like a BTK inhibitor or venetoclax, and just get hooked into the system early so that by the time they come for CAR T cells, they're not already 3, 4, 5 lines into treatment and not in good shape. That usually helps the patients a lot. And I think we've seen better responses, more durable responses in that situation. So right now we'll have to offer CAR T cells on a trial for CLL because it's not FDA approved for CLL yet. But I'm hoping in the future, as it becomes more commercially available, that we can start doing it in earlier lines of therapy for patients as well. So early referrals are very important.